Just before you start again, Mr. Hill, let me just say something to those who may have been watching, or should I say hoping to watch, on YouTube. I'm very sorry that it seems that your transmission was shut off. Let me explain why. The YouTube systems detected, so I'm told, that something which they thought was copyright was being displayed. And their systems are such that it instantly shut down the transmission. It was copyright, of course we had the right to play it, but that was why you didn't see what you may already have picked up from other sources, which is the transmission of the 1st of December 1975, from World in Action. We're back online now. If you haven't been discouraged from continuing to watch, I'm sorry once again for that, but I hope that's a sufficient explanation. It's what I've been told, and we're ready to begin.
Thank you, sir. Before we return to the narrative for Travenol and Hemophil, I'm just going to refer back to some evidence that we heard about Immuno, and in particular about Mr. Berry and what he said in 1979 about the difference between American plasma cryobilin and European plasma cryobilin.
What he was reported to say.
What he was reported to have said, precisely, sir. So if we could have, please, on screen, Cernic, HCDO 40403. This is page 3 of the minutes of the Haemophilia Reference Centre Directors meeting of the 6th of April 1979, the document that we looked at yesterday. The relevant section in particular being Professor Ingram has been in contact with Mr. Berry of Immuno, who has said that their action was aimed at making available to clinicians material which may carry less risk of transmitting hepatitis. If we could also have on screen, please, on a split screen, LOTH 5012_136. And I should say that I'm grateful to Ms. Gollop for bringing this to my attention. This is a letter written on the 8th of August 1979 by Professor Ingram to Dr. Ritzer, who had, as we understand it, prepared those minutes. What Professor Ingram says is, "Thank you for sending me the minutes of the 8th meeting of the Haemophilia Reference Centre Directors. At the top of page 3, I would like to ask you to expand the record of my reported conversation with Mr. Berry of Immuno. Would you please replace the latter part of that sentence with," and I quote now, and I understand this to follow on from the section where it said, "Professor Ingram has been in contact with Mr. Berry of Immuno," and the new version is, "who had said that the American blue material was offered for those who wished to take advantage of the lower American price, whereas the European red material was still available for those who felt that it carried a lower risk of conveying hepatitis, although the company regarded both products as equally safe." That is Professor Ingram correcting the minute which purported to record what Mr. Berry had told him. The only additional points I would make in respect of that is that the reference to the company it is unclear whether that is intended to refer to Immuno Limited, the UK company, or Immuno AG, the Austrian company, or both. And this record was in 1979. The document that we looked at, the internal Immuno AG document that we looked at yesterday, in which reference was made to the hepatitis risk of the two products, that was from 1976.
I thank you very much for that. It's important that should be there to stand beside the evidence that we've thus far heard. Is there any evidence to show that Mr. Berry was aware of the way in which the discussion at Immuno AG was recorded, where Immuno AG recorded a view which essentially meant that they would seek to license American product.
I have seen no evidence that Mr. Berry was aware of that discussion.
Yeah. Yes.
The other point that you raised, another point that you raised, so thank you, Senator, we can take that off the screen. Could we have instead CBLA 5011_005, page 9? And I'm grateful to Mr. Cummings for this. You asked about when it was that donations were pooled, as per the description that had been given by Dr. Kingdon of Hyland's processes. The answer comes in paragraph 22 of his statement, which follows on from the section that I read, which says that once the computerized inspection is completed, the donations are combined into a start pool. The context of that is that the donation has by that stage already been tested at Hyland's testing centre and has also been transported to Hyland's manufacturing centre, and there has been a period in which it has sat in quarantine at the manufacturing centre. It has then been processed on at least by 1990 when this statement was written, it had been inspected via a computerised line, which had been introduced in 1984 to 85, and it was only after that inspection that the different donations then get pulled together.
There was some reference, which you showed me earlier, about the greater difficulty of doing something manually. What?
If we could go, please, Sir Nick, to the previous page, page 8. It's actually the previous paragraph, paragraph 21. This is the section that we read earlier.
Yes.
The computerised inspection line was introduced in 1984 to 1985. Prior to this, the same procedures were undertaken manually. That is where the revised statement ends. The first draft, prior to Dr. Kingdom's revision, went on to complete that sentence by saying, although under the manual procedure, donations from donors who had seroconverted between one donation and another were unable to be removed.
So, in other words, until if that had stood, it would be saying that until the computerized line was introduced, then the material was liable to be pooled without a donation. The fact that a donation was infected or potentially infected being recognised, and that having the result of the donation itself being withdrawn. Is that what it's saying?
What I understood this to mean is that the computerised system allows the operator to run a barcode, and if the barcode flags up that that person has now tested positive for HIV, then previous donations can be pulled from a line if they still exist. Under the manual system, that stage wasn't possible.
I see.
But it may be that you could still pull that donation, but you just couldn't identify the other ones.
So it may be previous donations which haven't yet got to the stage of being pooled, or may have been pooled.
Yes. I stress that is my interpretation of the statement.
Well,
others may be available.
It's not clear. The true meaning may be a matter of submission in due course.
Yes. And as you said, sir, we don't know why that was struck through by...
No, but plainly he didn't wish it to be in for whatever reason.
Yes. Senator, can we have on screen, please, MHRA 304180. And we're returning now to the narrative. We can see that this document is entitled Note of a Meeting of a Divisional Management Group held on the 9th of December 1975. It is a DHSS group. We can see Dr. Holgate's name there amongst a number of other people who were involved in that meeting. If we could go to page 3, please, Sir Nick. under any other business. 9th of December, the day after the second of the World in Action documentaries had been broadcast, and 8 days after the first. Any other business? Blood coagulation factors for haemophilia. Mr. Tringham reported that a television World in Action film had made criticisms of Travenol's production of factor VIII in the USA. The criticisms were in conformity with an inspection report carried out on behalf of the division. The Minister of State had been briefed and was concerned about the supply of the factor and about the hazards of using it. I pause there to say that the Minister of State at the time was Dr. Owen. A similar product manufactured by Armour had recently been cleared by the Committee on the Safety of Medicines. Supply Division were anxious that it should be licensed as it would be available at a lower price than the Travenol product. There was some doubt as to whether the collection of blood products for either product was satisfactory. Dr. Holgate said that he doubted whether inspection of the American collecting centres would be useful. What was needed was to strengthen the requirements in the product licence and to insist on returns from each collection centre, including the rate of rejection of donors or donations. The story of the television film was that Britain could be self-sufficient in factors for haemophilia within a few years. The department in 1972 had allocated £500,000 and had bought the equipment needed for production. However, there were some difficulties. Neither of the production units had applied for product licenses. The SHHD, that is the Scottish Home and Health Division, have written to the factory at Liberton, and Medicines Division have reminded HS2 Division that though the Lister Institute of Preventative Medicine, Elstree, had a contract with the department, this did not give them exemption from the requirement to hold a product licence. Mr. Tringham emphasised that his concern was to regularise the situation, not to stop production. The haemophiliac patients groups were protesting at the delays in commissioning plant. Supply Division were concerned that in the meantime, doctors might develop brand preferences for the imported products. Dr. Holgate said that the doctors would prefer the British products as being safer. Indeed, once a pure supply is available, doctors will want to use the product in situations in which the currently available factors would be too great a risk. That is the end of that discussion. The reference to the Armour application is one that will be picked up next week in the presentation on Armour. So DHSC 3037191118, please. This is a document that we looked at yesterday, sir, and it's designed to show how much Hemophil was being used as of this time, which is the 21st of December 1976. So we've moved on another year down the line. The October sales in 1976 for the Travenol product, which we take to be Hemophil, were 865,680 international units. And overall, for the 12 months to the 31st of October 1976, a little over 5.2 million international units at a value of £627,738. It is, as we can see there, the most used commercial product at that time. On the 26th of August 1977, so again a leap forward of almost a year, Travenol submitted an application to vary the product licence in order to add the facility at Lezine in Belgium as a manufacturer and to include a correction to the test method used for screening each plasma donation for hepatitis B surface antigen. And the correction was that each plasma donation was tested using solid phase radioimmune assay procedure rather than counter-electrophoresis. The variation application was withdrawn on the 30th of January 1978, and instead it was said that the changes would be included in the renewal application for the license. And that's SHPL 40276 underscore 106, and the same stem underscore 081. The product license was renewed on the 19th of February 1978, SHPL 40283 underscore 005. We have looked at the 1980 annual returns before. I won't bring them up. I will simply remind people of the figures. As of 1980, the Royal Free Hospital treated patients with approximately 31,500 units of Hemophil, making it the least used of their commercial concentrates, other than a Spaywood product. By contrast, the Royal Free used more than 2 million units of factor VIII. Forgive me, I've gone on to the wrong product there. So Royal Free, 31,500 units of Hemophil. Royal Victoria Infirmary in Newcastle, 54,400 units of Hemophil, again, the lowest of all of its concentrates. Northern Ireland recorded more than 520,000 units of Hemophil, which made it second after cryobilin. As we saw yesterday, Hemophil in Northern Ireland was used for inpatients and patients with factor VIII antibodies, whereas cryobilin tended to be used in the home treatment. In 1979, Hyland and Travenol introduced a variant of Hemophil, which was called Hemophil 2. We haven't identified many documents about Hemophil 2. Those which we have identified show that it was on the market until November 1982, when it was taken off for commercial reasons. It was said to be unprofitable in light of currency variations in particular. The original Hemophil was classed as a high purity product. Hemophil 2 was an intermediate purity product and was intended to be sold at a lower price. But ultimately that didn't prove economic for the company, and that's why it was taken off the market. The reference is DHSC 302223 underscore 055, and then the same stem underscore 056, the same stem 058. The inquiry hasn't identified any documents indicating that Hemophil 2 received a product license. There is then a twist in the story which we frankly haven't got to the bottom of yet. On the 16th of December 1982, so we've moved forward several years and we're getting towards the end of a 5-year period under which the then active license ran. On December the 16th, 1982, Travenol submitted a renewal application for Hemophil, and they did it on a particular form. As per a request by the DHSS, they then resubmitted it under a different form, and it was called the Review/Renewal of Expiry License form. And that was done on the 26th of October 1983. Renewal documents were never sent back to Travenol, Travenol Limited this is, and we can see from the correspondence that they assumed that the department had renewed the product license under that new scheme. By late November 1984, however, Travenol Limited had been informed that the blood products were not covered by this scheme. So even though the DHSS had told them to reapply under that scheme, actually it didn't cover blood products. The Hemophil product license expired on the 19th of March 1983. So there is then this period between the 19th of March 1983 and November 1984 where there is a legal issue, I suppose, as to the status of Hemophil, as to whether it was a licensed product or not. The references are MHRA 309345, SHPL 40283 underscore 005, there is confusion there, and I don't seek now to pretend that we have got to the bottom of that or to attribute any responsibility for where that confusion lay. The document from the 28th of June 1983 that we looked at yesterday, which is the table that the DHSS compiled showing the answers that they had got to Dr. Walford's questionnaire, showed that Hemophil had an annual sale of 8 to 9 million international units, making it one of the most used Factor VIII products, but some way behind the leading product of that time, which was Factor VIII, which was somewhere in the region of 15 to 20 million international units. We've now reached the part of the narrative where we get on to the heat-treated product. As we know from Dr. Kingdon's statement, the American company had been pursuing heat treatment for some time by this stage. The first clinical trials had taken place in France in 1982. In March 1983, the FDA granted a license for Hemophil T in the United States. In April 1983, the West German authorities granted a license for Hemophil. And by the time that the UK application came to be made, which is what we will look at in a second, Hemophil T had also been licensed for sale in Canada, Spain, Sweden, Belgium, and Ireland. Reference for that is SHPL 40283_005, and also paragraph 51 and 46 of Dr. Kingdon's statement. In April 1983, Travenol Limited met with Dr. Fowler and Dr. Purvis of the Medicines Division of the DHSS to discuss Hyland products in general and regulatory issues, and it's in this context that they raise the heat treatment of Hemophil. If we could have on screen, please, Cernic, SHPL 40233_058. This is an internal Travenol Limited document, so a British company, and we can see its address in Norfolk in the top right-hand corner. We can see it is from Mr. Cameron to Mr. Chard, copied to various others, dated the 25th of April 1983, the subject, meeting at DHSS with Dr. Fowler and Dr. Purvis. If we could expand that, please, a little. Thank you very much. The minute records, C. Chard and A. Cameron met Dr. Fowler and Dr. Purvis of the Medicines Division to discuss issues relating to the Hyland therapeutic ranges and several other regulatory areas. The first thing they discuss are plasma protein fractions and buminate. I won't trouble you with that. Second, Hemophil T, at the bottom of that page. Doctors Fowler and Purvis had previously received details of this study and stated that they could not really comment on this product and we should submit the file and wait for the response. The final product license application would have to be seen at committee level, and although product costs should not come into the discussion, the overall cost benefit would be examined. I can't assist as to what was meant by that comment. If we could go on to the next page, please, Cernic. One issue that would have to be included in the final submission would be a quantification of hepatitis risk reduction on heating. We can see there, sir, another example of how the licensing process works. There are meetings between DHSS officials and the companies, and the DHSS officials tell them the kind of things that they would expect to see in a license application. Overall conclusion, the meeting proved very useful, and Doctors Fowler and Purvis made many constructive suggestions as to future regulatory approaches for new products. Then over onto the next page, please, Cernic. One point that was raised by Dr. Fowler was the increasing use of release on prescription only as a means of marketing products without a product license. This issue has been raised at a very high level, brackets Secretary of State for Health, and has occurred because of large figures appearing on health authority budgets for products which are not licensed. The DHSS considers that the prescription-only system of release is being misused by several companies as an alternative to obtaining a product license. There is a considerable likelihood of the system being tightened up to prevent continued abuse by large companies. That takes us back, sir, to a comment you made yesterday about why it was that the 1984 order was made and the motivation behind that. And this document perhaps gives us some insight into that and why the system was, why it was felt needed to tighten the system up. In May 1983, Travenol submitted a variation to the product license for Hemophil to incorporate a dry heat treatment step at 60 degrees for 72 hours in the final stages of manufacture, so that is Hemophil T. The CSM Subcommittee on Biologicals assessed the application in September 1983, assisted by a report prepared by Dr. Fowler and Dr. Purvis. And if we could have on screen, please, DHSC 0105556_028. We can see the names of Dr. Fowler and Purvis there, the same people who had met with representatives of Travenol Limited in that previous document that we looked at. The application was received in May 1983 to be considered at the September 1983 meeting of the Committee on the Safety of Medicines. If we could now go to page 5 of this document, please. This is part of the report that was produced for consideration at those meetings. And this is from the pharmaceutical, of a pharmacist providing a pharmaceutical comment. If we go down to point 5, there are various technical observations about the application. And it says that it is noteworthy that, in point 5, no reason has been given for the inclusion of the heat treatment step. This must raise questions about the justification of such a treatment in view of the fact that up to 20% of the activity of the product may be lost by virtue of its inclusion. This could lead to the product containing up to 20% of degradation products. Note, if it has been included to reduce the chance of transmission of viral infections, one would have expected to see some work done with a product spiked with known viruses. Anecdotal information indicates this step has been included to minimize the chance of transmission of AIDS, but this assumes AIDS is a viral mediated infection. There is no evidence to confirm this. We could then go on, please, to page 7, to the medical comment. This says, the company have applied to vary their product license for a conventional factor VIII concentrate by the addition to the manufacturing process of a heat treatment step. No reason is given for this, but it may be assumed that they have a reason because the proposed treatment destroys about 20% of the coagulant activity yield. 2 letters sent by the company to regional transfusion directors and specialists in haemophilia, together with the reprint from Hospital Infection Control for May 1983, provide the answer. See appendices 5, 6, and 7. This application concentrates on the coagulant's properties remaining in the product after heat treatment and skates over the toxic potential of degradation products associated with the loss of 20% of the coagulant activity. The brief summaries of results obtained from animal and human studies are inadequate for the purpose of assessing this variation. It will be seen that the reports from in vivo animal studies are conclusions rather than results. There are no data. Although the reports of studies in humans are more forthcoming with regard to data, they are still seriously lacking in experimental and clinical detail. Numbers are small in all the in vivo studies, both animal and human, so even if a full detailed description of the work had been provided, it might still have been thought insufficient to support this variation. Medical recommendation, but this variation be refused on the grounds that the evidence of safety and efficacy was inadequate. Remark, the committee may feel that on the evidence of the 2 letters sent by Travenol to transfusion and haemophilia specialists, that the company are already promoting heat-treated haemophil as being less likely to transmit viral infection, with particular reference to hepatitis B, non-A, non-B hepatitis, and possibly AIDS. The inquiry, sir, hasn't identified all of the appendices to which reference is made there, but we have found one. PRSE 304496, please, Sir Nick. This is a letter dated the 9th of May 1983, which was sent to the Department of Health and Social Security. It says "Dear," and then a name has either been removed or was not included. And it is possible that this was a circular that was sent to others as well, and that's certainly the impression that one gets from the medical comment that was made. The letter says this: I want to advise you of important developments and actions being taken by Hyland Therapeutics and Travenol Laboratories in connection with the risk of acquired immune deficiency syndrome, AIDS. While the causative agent of this disease remains to be identified, some evidence suggests that it is caused by a virus that can be transmitted by blood and certain blood products. The letter goes on to describe some of the steps that Hyland had taken in response. It also describes how a donor had been identified, although not finally diagnosed, as having been a possible victim of AIDS, and the steps that Hyland in the United States had taken in response to that. And it was stressed that no therapeutic products fractionated from plasma pools that contained this donor's plasma have been shipped to any customers in Europe. If we go over to the next page, please, Sir Nick. Hyland described the screening procedures that they have put in place, and then they say this in the second paragraph. In addition to screening procedures to eliminate high-risk donor groups and placing in quarantine all products made from plasma pools affected by this donor, Hyland is taking a third major action it believes could contribute to the safety of the haemophiliac. Hyland will, as expeditiously as possible, convert both its European and US facilities to manufacture only heat-treated factor VIII products. This new heat-treated product, Hemophil T, which Hyland Therapeutics has recently introduced, has equal potency and effectiveness as normal hemophil, but has been subjected during manufacture to an additional heat-treating step designed to reduce active viral content. This new product is being offered at only a small price premium over the regular non-heated product. Since the causative agent for AIDS has not been identified, and since the effects of the heat-treating process on all viruses have not been determined, Hyland Therapeutics cannot, at present, give assurance that the heat-treated product eliminates the risk of transmission of AIDS. However, Hyland Therapeutics believes that administration of the heat-treated product, designed to reduce active viral content, may increase patient and centre personnel safety. I won't read the rest of the letter, sir, but that is one of the letters that have been identified as being problematic by those who were assessing the Hemophil T application. The Committee on the Safety of Medicines Biological Subcommittee concluded that they could not recommend that the licence be granted or be varied. Could we have on screen, please, Sir Nick, DHSC 303951 _006. These are the minutes of that meeting as they referred to Hemophil. They are as most such minutes, relatively short. The recommendation is that on the evidence before them, the subcommittee, on grounds of safety, quality and efficacy, was unable to recommend that the product licence should be varied as indicated. The subcommittee considered that 1, justification should be provided for the inclusion and choice of the heat treatment step. 2, the heat-treated product was inadequately characterised. 3, inadequate evidence of safety and efficacy was provided. 4, in the event of a grant of a variation to the licence, labels and data sheets should be modified to the satisfaction of the Secretariat. We could expand the remarks. 1, promotional letters making unjustified claims on improved safety margins in respect of infection and AIDS were seen by the subcommittee and strongly deprecated. 2, evidence of the long-term safety in haemophiliac patients of treated products such as this is regarded as an important prerequisite of licensing. Travenol were notified on the 14th of October 1983 that the variation application had been refused by the Committee on the Safety of Medicines on the grounds that were listed above as SHPL 40283_005. Travenol Limited submitted an additional variation application for Hemophil in September 1984 to include a heat treatment step. In November, before I move on to 1984, so I'd just make the point that this was all taking place in October 1983. Yesterday, when we were discussing Immuno and the documents that we saw encouraging companies to apply for heat-treated licenses, that was taking place in the autumn of 1984, so a year later. It is in that context that we move on to September 1984, when Travenol Limited submitted a further variation application for Hemophil, again to add the heat treatment step, and that is SHPL 40283_005. In November, the DHSS requested that Travenol instead submit an abridged licence application for Hemophil T, SHPL 40283_005. And as we know, by that time, the DHSS were encouraging all companies to put forward such applications. On the 30th of November 1984, Travenol submitted an application for the abridged product licence. That's SHPL 40283_005. And I will show one page from that application. Could we have that on screen, please, Sir Nick, and page 10 of that document? This is part of the application, and this is, as people will no doubt recognise now, part of the warning section of the data sheet that would be put forward. And it says, warnings, this concentrate is prepared from large pools of fresh human plasma, which may contain causative agents of viral hepatitis. However, each unit of plasma used in the manufacture of this product has been found to be non-reactive for hepatitis B surface antigen when tested with licensed third-generation reagents. In addition, this product has been subjected to a heating procedure during its manufacturing process designed to reduce the risk of transmission of hepatitis. Although these testing and heating steps reduce the risk of hepatitis transmission, the possibility of such transmission should be considered in use of the product. There is, as you will see, so no reference to HTLV3 or LAV or AIDS as part of that warning section. A study was submitted with the application. This can be found under the same reference. And that study referred to, was one of the studies referred to in Dr. Kingdon's summary, and it's the one that was carried out across 10 haemophilia centres in 5 countries in Europe, including in the UK, under Dr. Savage and Dr. Arenstam. At that stage, the study was reporting that 55% of patients had developed post-transfusion hepatitis. As we know, the figures change as the results continue to come in. In each case, it was classified as moderate non-A, non-B hepatitis. The application also reported 8 adverse reactions to the use of the product, some of which related to non-A, non-B hepatitis, 4 in fact. The company placed this in the context of an estimated 4,000 plus patients having been treated with the product, and more than 190 million international units having been sold. That's a worldwide figure at that stage, and we've seen that it's licensed in other countries. The company argued, and I quote, "The reported rate of adverse reaction is low," and also argued that, quotes, "non-A, non-B hepatitis is a common condition in haemophiliac patients on factor VIII therapy." The licensing authority granted Hemophil T a product license on the 27th of February 1985. The reference for that is CBLA 606_023. On the 3rd, so that's February 1985. In June 1985, Travenol Inc. withdrew non-heat-treated Hemophil and non-heat-treated Proplex from the US market and replaced unused units with heat-treated units. That's CGRA 40472. That's the US market. We are still investigating what happened in the UK market, and we'll come back to that in November. There was a further variation application on the 13th of February 1987 to include a specification that the product was manufactured exclusively from individual plasma donations screened for HDLV3 antibodies as well as for hepatitis B surface antigen, and that the plasma was obtained only from donors with normal ALT levels, SHPL 40282_004. I note again, similar points were made earlier, although that application is coming in the 13th of February 1987, some of those steps at least have been taken some time before that. I'm about to move on, sir, to the next generation of Hemophil, which is Hemophil M, a monoclonal purification method. I note the time, sir, and I wonder if that is a convenient moment.
So when are we going to complete the story of Travenol and Hemophil?
On Tuesday.
On Tuesday, starting at 10.
I'm in your hands on that, sir.
Well, we'll start at 10. So 10 o'clock on Tuesday, and that's our next sitting.